Hello, my name is Breitschwerdt. I'm the Melanie S. Steele Professor of Medicine and Infectious Disease in the Comparative Medicine Institute at North Carolina University College of Veterinary Medicine. In the context of disclosures, I, in conjunction with Dr. Sushama Santaki at North Carolina State University, hold a U.S. patent on the cultivation of microorganisms in an insect biochemical composition growth medium. I'm the co-founder, shareholder, and chief scientific officer for Galaxy Diagnostics, a company that provides advanced diagnostic testing for the detection of Bartonella species. I do not intend to reference any off-labeled or non-FDA approved usage in this presentation. So our learning objectives for this module are to focus on limitations of Bartonella species serology for the diagnosis of Bartonelloses, to describe the limitations of conventional PCR versus quantitative PCR versus droplet digital PCR for the detection of Bartonella species DNA in patient specimens, and to describe the diagnostic utility of using an enrichment culture approach in conjunction with droplet digital PCR for the documentation of Bartonella species infections. So the place we need to start with diagnosis in Bartonella is prior to 1990, we didn't have to worry about it because we did not know these bacteria existed on the North American continent. And then in 1990, as we have discussed, all of a sudden we know that there are three species, Bartonella henselae and Bartonella Quintana in HIV-infected individuals, and Bartonella bacilliformis, which was known for many, many years in the Peruvian Andes. But what subsequently changed over the next ensuing years is the fact that researchers around the world kept finding more and more Bartonella species associated with an increasingly diverse group of reservoir hosts. And those that are highlighted here are the ones that have been associated with human infections and disease. And the others at this point that are in yellow have not. But we now have exceeded 36 species, and it's a little hard to keep up with the count, even if you're a Bartonella researcher. But I think my current estimate is we're pretty close to 42 named species of Bartonella. So one of the important things that we all know is that all diagnostic tests have inherent limitations. And although we frequently throw around the term gold standard diagnostic tests, most of these tests that we used are not made of gold. They are closer to tin than they are to gold, perhaps. And that's really important because what it should always emphasize is that our clinical suspicion is absolutely critical in regard to the interpretation of any diagnostic test result. And I think the other emphasis for all of us as clinicians is that we need to at least understand those big limitations associated with the test so that we can better interpret the test for our patients. And for many infections, as we've discussed previously, disease occurs as a result of immunological imbalance. And when that imbalance occurs and the infectious agent persists, what we often see is an immune complex disease, such as immune complex glomerulonephritis, or an autoimmune disease, such as Guillain-Barré syndrome, where antibodies would be directed against myelin-basic proteins on the lower motor neuron, which can occur in association with vaccinations or Campylobacter infection. And we have seen that in association with Bartonella as well. So again, a spectrum of cross-reactive antigens that ultimately induce an antibody that damages part of an organ system, be it the kidney or the nerve. Concurrent infections, as we have emphasized, are more common than is currently recognized, and concurrent infections are really complicated in as much as one organism may predominate and the other organism, which might be in the same genus, may be there in very, very low concentrations. So documenting co-infections of different organisms or the same organisms really complicates the job of the medical microbiologist. So in a previous module, we emphasized this context of frontal pathogens and stealth pathogens. And here I want to emphasize the fact that frontal pathogens are often readily diagnosed because there is a very rapid replication. So within a matter of days, there can be very large numbers of influenza virus particles. The initial infections, oftentimes using PCR or a DNA or RNA detection, is far more sensitive than using serology because the onset of illness is so rapid that you can detect the organism before you can detect the antibody response. Whereas with chronic infections, we would like to think that we could detect the antibody response for a much longer period of time. But the disadvantage we have here is we can't document seroconversion. We can, if we treat and we have a positive antibody titer and we follow that titer, document seroreversion. So I'm just introducing this again to emphasize the fact that the diagnostic approach I would use for a patient that I suspect has an acute infection, whether it's influenza or Rocky Mountain spotted fever, would be very different than a patient that I suspected had ehrlichiosis, bartonellosis, babesiosis as a very, very chronic infection. So in the context of limitations for diagnostic tests, and I'm going to use specifically Bartonella as an example here, we routinely do blood culture when we suspect Bartonella bacteremia, and Bartonella endocarditis would be an excellent example. But because of that extremely slow dividing time, it can take anywhere from 2 weeks to, I think the record is 6 weeks, before the microbiologist can actually see bacterial colonies that are large enough to visualize on a blood agar plate. We also know that both in blood and cerebrospinal fluid, if Bartonella is the cause of the disease process, we are trying to identify an extremely low bacterial load. And this becomes the rate limiting factor that there are so few bacteria that trying to transition them from blood to a microbiological medium that will grow them up to a level of detection can be extremely challenging. And so it's not that they're not there, they're there in low numbers, but even that next step of transitioning their growth to a new environment becomes challenging in the laboratory. And certainly if antibiotics have been administered prior to attempting that culture, that will further compromise, as we all know, our ability to make an isolate or identify the organism. So serology, if it's positive, is great. One of the things that we will emphasize the fact that in both dogs and people, our experience is with Bartonella henselae as one example, up to 50% cannot have antibodies to the bacteria that we can identify in their blood. So in contrast to some other infections, seronegative infection with Bartonella, for reasons that remain unclear to me, is extremely common. And if we can identify antibodies in a patient, that doesn't prove that they are currently infected. It only indicates that they've been exposed and they may be infected. And it's the reason that we believe that combining indirect testing, where you're looking for the host immune response, and direct testing, where you're detecting an antigen, which can be done through a serology test such as an ELISA, or DNA or RNA to document active infection, is really, really critical. But finding that DNA, as we've alluded to, in a low bacterial load is a real problem in regard to sensitivity. So what we're going to emphasize towards the end is what we've worked on for years, and we believe for at least the genus Bartonella becomes the current gold standard, is combining enrichment culture with droplet digital PCR. So this brings us back to where we start with a patient that we're going to obtain specimens to look at diagnostically. And one of the things that we are clearly seeing if you're using direct detection is that tissue, if you can obtain a tissue from a diseased area or adjacent to a diseased area, you are more likely to be able to document Bartonella DNA than if you were to test blood or a pleural effusion or cerebrospinal fluid or a joint effusion. It's not to say that those other specimens cannot be tested and cannot be positive, but it's to say that these bacteria are intracellular and they localize in the area in which disease is occurring, and they're not always in that diagnostic specimen that is most easy for us to obtain. So what's available for the diagnosis of Bartonella? In the context of serology, commercially, there's IFA test, ELISA test, and Western blotting test. In regard to culture, routine culture on agar plates is rarely successful. It can be, and it's particularly if you've got an infectious disease physician like Dr. Jane Kohler, and you're dealing with immunocompromised patients, you can actually go to the bedside, obtain the blood, and put it on a blood agar plate and grow Bartonella if you incubate it under the right conditions and for the long enough period of time, which I've mentioned can be anywhere from 2 weeks to 6 weeks. It's also, if you're trying to get Bartonella henselae from that patient, you should use blood agar and as freshly poured blood agar plates as possible, because the older they are, the less likely the bacteria seem to be able to grow on them. But if you're looking for Bartonella Quintana in your patient, you should use chocolate agar where they preferentially grow better. So you can see already that we've got, even in routine culture, real challenges. And when we go to enrichment culture, there are additional challenges, but at least this one has become the one that gives us the most satisfaction in being able to say Bartonella yes or no in patient samples. So we're going to talk in detail about conventional PCR versus quantitative PCR versus droplet digital PCR. All these modalities are available and used with Bartonella at this point in time. Immunohistochemistry is relatively sensitive. Again, you're looking at diseased tissue using an antibody to light up the organism. Unfortunately, there was only one commercially available monoclonal antibody for Bartonella henselae, and that was the only Bartonella species for which we had a monoclonal antibody, and the company is no longer making that antibody. So immunohistochemistry, until there's more emphasis on the genus Bartonella, is not available. And then FISH, fluorescent in situ hybridization, is identifying a specific chunk of Bartonella DNA that we develop a probe that'll set down on that and allow us to visualize the organism. So all of those tests are available in various laboratories for the diagnosis of Bartonellosis, and they all would vary in regard to specificity and sensitivity amongst laboratories that are doing that testing. Now, for this particular module, I am again going to emphasize the fact that dogs seem to be the best naturally occurring model for human Bartonellosis, and humans seem to be the best naturally occurring model for dog Bartonellosis, because one, they develop the same disease signs or symptoms, but two, immunologically, they are showing us the same patterns in the same picture in regard to what we're seeing in serology and what we're seeing in regard to isolation or enrichment culture, identification of these bacteria by PCR testing. So using dogs and humans as our example, this is a publication by Dr. Nelson from the CDC in Emerging Infectious Disease in 2016. And this is a publication by Dr. Aaron Lashnitz in our laboratory in the Journal of Veterinary Internal Medicine in 2018. You can see that we are talking about those nearly 16,000 samples evaluated at the vector-borne disease lab submitted by veterinarians. These would be human cases from the human database of diagnosis of cat scratch disease, meeting CDC criteria for that diagnosis. And I think without going into great detail, what we see is you can get cat scratch disease anywhere in the United States. And although the prevalence varies, and clearly this is the area in which there are fewer fleas and likely fewer other vectors, even including ticks to a certain extent, that could be vectors for Bartonella, that we've got 6.4, 4.6, 3.5, 3.7 per 100,000. And Aaron's results are very similar. Is the dog is literally going from where we didn't have enough samples to make any association up to about 6 or 7%, but saying that a dog can have antibodies to Bartonella anywhere in the United States. So this is some very recent work from our intracellular pathogens research lab, and we decided to work with a company, IDEX Laboratory, to look at the feasibility of using ELISA versus IFA. And it was very interesting because the overall patterns that we saw in regard to finding infected dogs throughout much of the United States was very similar. But what's depicted here, and I realize this is somewhat of a complicated slide, so we're just going to talk about a couple things here, is that if we did IFA, what you're going to see is a large number of dogs that only had antibodies by IFA to Bartonella cholerae, a large number that only had antibodies to Bartonella henselae, and a relatively small number that only had antibodies to Bartonella binsonii burgdorferi. And then this whole group had antibodies to multiple Bartonella species. And we'll come back to this as why it's important in our experimental infection studies in just a minute. But contrast that to this particular slide, where we go from BK here to 4 out of 362 here. And so this was a very different pattern where binsonii burgdorferi by IFA was more prevalent than henselae, which was more prevalent. So all this says to us is, and these are, in fairness, different populations. These were sick dogs that were tested diagnostically because veterinarians suspected a disease. These, on the other hand, were all dogs sent to a very large commercial diagnostic laboratory and probably included a substantial number of dogs for which veterinarians were doing annual or every 3-year blood work as part of their geriatric screening as these dogs aged. So it's not a head-to-head comparison, but I wanted to use it to emphasize that these are all the same isolates as we used here in IFA, and these are all whole cell antigens rather than specific peptides. And as we look at these two, the results that we've gotten are very, very different. So what can we say about IFA serology? Is it's widely used, it's indicative of exposure, and its specificity is questionable. And I'm going to show you that in dogs, it's not quite as questionable as it is in humans at this point in time. The limitation is it's hard to standardize among laboratories in regard to what constitutes a cutoff value and how that's established. In humans, there is potential cross-reactivity been reported with Coxiella, Toxoplasma, and Chlamydia. But there's also studies in which there's no cross-reactivity to these organisms in people with known Bartonella infections, suggesting that sometimes it may have been that these individuals had been exposed to these organisms, and that was the reason for the reactivity rather than cross-reactivity to Bartonella antigens. Various epitope expression due to genetic rearrangement. So these organisms change over time during infection in laboratory studies and in non-human primate studies. And alteration in the sensitivity is dependent on the strain and the species, whether you're using cell culture grown organisms, which we routinely use in all our research and diagnostic testing, versus a non-cell culture cultivated organism from a blood agar plate, and how often that bacteria has been passaged from the time it's initially isolated, because an organism that's been passaged multiple times is likely been changed in regard to its surface protein expression and how it would be used. So in regard to specificity, we elected to infect 2 dogs, one with Bartonella henselae, which will be this dog, and another with Bartonella cholerae. These 2 dogs were part of a genetics colony, and they were heterozygotes, so they weren't being maintained in the colonies, and they were being adopted out for other studies. And what was fortunate or unfortunate, depending on how you look at it, is that we figured out just a couple days before we were going to infect with Bartonella henselae and Bartonella vinsonii burkholfi that both of these dogs were not only seropositive to Bartonella cholerae, but they were also blood culture positive to Bartonella cholerae and PCR positive. So we ended up by chance with 2 dogs that I had to make a decision: are we going to go ahead with this infection study, or are we going to not go ahead with the infection study? And we decided to go ahead, which was really interesting from a number of standpoints. So we put in Bartonella henselae, which would be this guy here and this color here. So you can see all of a sudden we've got antibodies to the Bartonella that went in at the highest concentration. And again, realize that all of these are very, very low antibody titers that we are seeing in this instance, because these organisms are relatively non-pathogenic in dogs. And then the only other one that we're seeing positivity to is another Bartonella henselae strain, this purple here. And as you look down here, initially the purple was negative, it was only cholerae. Then henselae SA2 pops up, then Houston 1 pops up. But throughout this entire study, vinsonii burkholfi, and there are 3 different genotypes that we can separate very readily by intergenic spacer sequencing and other means that literally could be different species, but are considered different subspecies. So you can see that none of these are ever recognized. The other thing that was going on is we were unable to document infection during this period, so we gave dexamethasone at an immunosuppressive dose, hoping that we would then be able to document bacteremia, which we were not. But what's also striking is this rapid drop-off that occurred after that in both of the dogs in regard to their recognition to all of these different antigens that we're testing against. And I think without having real data to be able to prove this, it seems like the longer individuals are infected, the more likely they are to become IFA seronegative in a state of chronic infection. So this is the second dog infected with Bartonella vinsonii burkholfi. And in this instance, again, what you can see is we've got Bartonella cholerae antibodies and only Bartonella cholerae antibodies. And then what happens next is we get Bartonella vinsonii burkholfi, the strain that we put into the dog, and then we start getting antibodies to the other genotypes of Bartonella. Then we give dexamethasone, and I'm not sure what exactly happened here, but this was repeatable. And what you can see, what we're left with is the Bartonella Vinsonii burkholfi genotype 3 that we infected with and Bartonella cholerae that the dogs were initially infected with, and then everybody goes negative again out through 89 days. Both of these dogs were ultimately, by tissue PCR, proven to be infected. So let's now go from dogs to humans, and this is a study that was presented at the 4th International Conference on Bartonella's Emerging Pathogens. And the study was done in Sweden, and what these investigators did was they essentially took serum from a large number of individuals that had had cat bites and tested in their laboratory. And then anyone who was seroreactive to any of the strains, these 4 strains down here, at a titer greater than or equal to 1 to 64, was included in the comparative study. So a total of 58 patients were included. And what this pie diagram illustrates is that if they tested against a Swedish cat isolate in people that were bitten by a cat in Sweden, they could detect antibodies, IgG antibodies, in about 48% of them. But if they were to use a French strain, the Marseille strain, or the Houston 1 strain, which is the American tissue culture type collection strain from the United States, they would have gotten 14 or 17%. So it is another example to illustrate that all these people were negative to the strain out of a cat in Sweden that identified most of the Swedish patients that were bitten by a cat and seroconverted to a Bartonella henselae strain type. So going back to this idea of Bartonella IFA specificity, we've spent a lot of time on this in multiple publications, and my bias at this point in time is that if we experimentally infect dogs with Bartonella Vinsonii burkholfi, they make genotype 1, they make antibodies to genotype 1, and they do not even make antibodies to the other genotype, much less Bartonella cholerae or henselae, excuse me, or Quintana, which we've looked at and is not on this slide as well. And again, this is the study that I showed you previously, where the dog was infected with Vinsonii burkholfi genotype 3 and made antibodies only to that one. That dog, the star means that dog was infected with cholerae to start with, so it had antibodies to cholerae. Same down here for the dog with SA2, which did have antibodies to the Houston 1 isolate, the HACC Houston 1 isolate. And then with a flea study where we infected with a Houston 1 type isolate, referred to as the CSU or Colorado State University 1, it's a strain that genetically is similar or identical to the Houston 1 isolate. Again, we did not get seroreactivity to Bartonella henselae SA2 or to any of the others. So the good news about IFA specificity, if you're looking at IgG, is that it's very specific. The bad news, as we've alluded to in this module and others, is it is not sensitive and can miss literally up to 50% of patients that are ultimately shown to be PCR positive. So I'm very proud of the fact that our research group has now provided 2 major innovations to facilitate Bartonella species diagnosis. And the first one, which is the patent that we generated for BAPGM, or this Bartonella alpha proteobacteria growth media. The second, thanks to a recent grant we received from the Cohen Foundation, is now the availability of Droplet digital PCR as a more sensitive PCR methodology to determine whether we can detect or not detect Bartonella DNA. So just a little bit on the BAPGM side of the story first, and then we're going to consider qPCR and ddPCR. So this is Dr. Santaki, and you'll see this is Christmas 2003, and you will also notice that I had a bit more hair, and fortunately some of it was still black. But you can see we had a very busy Christmas trying to sort out both in clinical specimens as well as inoculated specimens of Bartonella, how this media would behave. And so the principle here is that if we obtain a specimen from a patient and send it to the laboratory, and in this particular EDTA blood tube, there's actually 2 bacteria, and we extract DNA from that, which will be 25 microliters, then what you'll see is we may do an extraction that has no bacteria. And because we didn't test this entire thing, we couldn't. The techniques and the methodology will not allow us to do so. So there is always this risk of a false negative. And clearly what I tell our medicine residents and students and veterinarians in lectures is a negative PCR has never, never, never ruled out the presence of an infectious agent. It is only indicated that the laboratory did not amplify DNA or RNA from the sample that was submitted during the testing that the laboratory did. Why we became very excited about this insect biochemical based optimized growth media is the fact that we could take and go from just these 2 bacteria to a larger number of bacteria. And then if we took the same quantity, which we're always limited by this amount that we can extract DNA and do PCR, again, same as this up here, then we're more likely to get PCR results. And if we're doing conventional PCR and quantitative PCR, we can actually take that DNA that we've amplified, sequence it, and establish what strain it is. So we refer to this as the BAPGM Enrichment Blood Culture Platform. It was actually published in regard to its validation in 2007 by Ashley Duncan and Ricardo Maggi when Ashley was a PhD student in the lab in the Journal of Microbiological Methods. It's a very complicated platform in as much as we're taking a sample, extracting DNA, and doing PCR. We're putting that sample in BAPGM, and at 7, 14, and 21 days on a research basis, we're again extracting DNA and doing PCR. And at 7, 21, and 14 days, we're subculturing onto plates, hoping to get an isolate, extracting DNA and doing PCR. If we get a positive PCR result by qPCR or ddPCR anywhere along the way, we then sequence the amplicons so we can tell you this is Bartonella vinsonii subspecies Burkholfi, genotype 3, genotype 1, or what have you. And this is why that approach is important. So this is published in Parasites and Vectors, the 13-year-old daughter of a veterinarian who went from being a straight A student to having problems with sleeping, essentially insomnia, problems focusing in her class, and problems communicating with her parents, literally. And we ended up entering her into our IRB study along with her father, who was also having neurologic problems at the time. And what you can see here is that every sample that we got was negative, whether we did blood, serum, or both, by that direct DNA extraction. But when we went into enrichment culture, we could pretty much consistently get Bartonella vinsonii Burkholfi, and we could get enough DNA to send that to a commercial laboratory that generated a sequence for that DNA, sent it back to us, which we analyzed, and it was Bartonella vinsonii Burkholfi genotype 2. You will also note that we and everybody else in the world have a tremendous problem going even from our enrichment culture, where we're pretty sure we've got plenty more bacteria now, to getting these things to transition from this liquid insect-based growth media to a agar plate where the colonies will grow up. But in this instance, we ended up with an isolate. So that's the utility of the BAPGM platform. But I think the concept we all need to come away from is the idea of an enrichment approach to detect pathogens of low bacterial concentration, regardless of the approach the laboratory is going to use. And in this instance, Dr. Michael Kasoy at CDC in Colorado used cultivation in vero cells. So instead of a liquid media, he used a cell-based assay with human serum as the growth media for these cells. He targeted a different gene target of Bartonella. We used the intergenic spacer region, which Michael has subsequently said is probably the best workhorse, as we have believed. But this GLTA or synthase gene really does preferentially and more effectively amplify the rodent Bartonella species than our ITS primers. Again, a difference in laboratory testing that is of importance and one that we do the best to address. But if this was simple, we wouldn't be having this particular lecture. So what Michael's group demonstrated is that febrile humans in Thailand, when they had frozen back blood clots, he could amplify Bartonella from 7.7% of those after co-cultivation with vero cells. So after using an enrichment step, he could do that. Prior to an enrichment step, all these were PCR negative. And again, all of these were rodent Bartonella species. Subsequently, Dr. Bai in his laboratory demonstrated using our BAPGM, which we provided to them for research purposes. She tested stray dogs from Thailand from the same area in which these patients were evaluated. And much to our surprise, because it's rare that we get a rodent, we've gotten Bartonella elizabethae out of dogs, and but of these, that's the only rodent Bartonella other than one other Vinsonii oropensis that we've gotten out of dogs. So what you can see here is that a lot of dogs in Thailand, probably somewhat in contrast to even dogs in the inner cities of the United States, where there might be a large rodent population and a large stray dog population. This is pretty amazing that she came up with 31.3% of dogs being infected in the same area and with essentially the same species. Again, this represents Bartonella Quintana, which we and others have also documented in sick dogs. So again, emphasizing our use, after we went through the validation of the platform, we started using the BAPGM platform clinically at North Carolina State University Veterinary Hospital. And this is not a random population of the dogs or a serial testing of dogs coming through our hospital. These were dogs that were pretty much handpicked by myself and another internist that had immune-mediated disease, positive anti-nuclear antibody titers, polyarthritis, immune-mediated thrombocytopenia, immune-mediated hemolytic anemia. And I mention that only in as much as if we tested every dog coming through NC State's hospital sequentially, we'd probably drop this down to 1%. But here we had 61 dogs, or 9.2% infected. And of those that we were able, through enrichment culture, to document their infection, 49% were Bartonella henselae, 28% were Vinsonii burkholfi, and 3% were Bartonella cholerae. Now, I've made the point in previous modules, and I want to make it again, that I believe these are the three most important Bartonellas that infect dogs and people in the United States of America. And obviously, there are diagnostic tests out there that have been developed for this one, but the other emphasis has been Bartonella Quintana because of the historical importance of Quintana and because it was also found in HIV-infected individuals. Quintana is still important and remains important, particularly probably in indigent homeless people throughout our country. But there are far more people, including some of the homeless, that are sleeping with their dogs than are infested with human body lice. And again, I want to emphasize the fact that in dogs, if you do enough testing and you target enough Bartonella genes, you can prove co-infection in 18%, which was part of the beginning of this lecture. And finally, that seroreactivity was extremely low in these dogs, with only 25% of the BH dogs having antibodies to Bartonella henselae and 50% of the Winstonii burkholzii being seroreactive. So this is one more example of an individual that had cat scratch disease. And I think, again, it's very likely that she did not eliminate Bartonella henselae after she was infected, because starting about a year later, she developed minor motor seizures and then progressed to generalized seizures and eventually became a patient at Duke Hospital. You can see, and I wanted to include this because, again, the justification for doing serologic testing and doing PCR testing from blood and doing enrichment culture. Here, for whatever reason, enrichment culture let us down, whereas there was enough DNA and we were fortunate enough to be able to amplify Bartonella henselae Houston 1. You'll see that 31 days later, she went back over to Duke and they provided another set of samples, and she is now positive in her blood, positive in her enrichment culture, and we ended up with an isolate. So all is good. And her antibody titers are actually almost like she's seroconverted, which I'm not sure I believe, but again, antibodies to all three organisms here. And these were actually being done, we were collaborating with William Nicholson at CDC. So these antibody titers are not from our lab by IFA, but actually performed at CDC, and this Winstonii burkholzii was using our genotype 1 isolate. So she kind of got lost to follow up, and there was the physicians who were taking care of her at this point in time elected not to treat her, and by 256 days later, she had progressed to generalized seizures and was back at Duke. Unfortunately, Duke was supposed to provide us with blood for enrichment culture and CSF for enrichment culture. We only got CSF and we didn't get blood. So regrettably, we don't have blood results at the same time the CSF results. But once again, you'll see that there was probably an extremely low number of Bartonella in the cerebrospinal fluid. Direct DNA extraction was negative, enrichment culture was positive, and by DNA sequencing, we've got exactly the same strain throughout an illness that now spans at least 256 days. She was subsequently treated and had resolution to a great extent of her seizures, obviously maintained on anticonvulsant therapy, and underwent seroreversion back to very low titers. And I think if we would have followed her long enough, she would have probably remained negative. And this is one more example, just again, to put the whole story together and to illustrate the complexity of serodiagnosis and molecular diagnosis after an acute infection with a stealth organism. So this is a veterinarian that was examining a fractious dog that had developed a large number of rapidly growing cutaneous tumors. Historically, this dog had been treated about 6 months earlier for ehrlichiosis. And after the veterinarian received the needle stick by doing aspiration cytology, these cutaneous masses, the hospital administrator asked me, was it possible that Ehrlichia was transmitted? Histologically, this was a histiocytic type of neoplasia, and unfortunately, the dog was discharged and the owner elected euthanasia, and there was no follow-up. But this was the question. And because 33% of E. canis seroreactors in the southeastern United States are concurrently seroreactive to Winstonii burkholzii, we entered her into our IRB study, tested her by IFA and PCR for Ehrlichia and for Bartonella. She remained negative for both antibodies and PCR on numerous sequential samples, just a subset of which I'm showing you on this slide. And this slide illustrates what we figured out about Bartonella after an acute transmission from a needle stick. Obviously, she had been perfectly healthy at day 5, which was the first sample we got post-inoculation from the needle stick, negative, negative, and no detectable antibodies. By day 33, we're at that point again that she was lucky we got Winstonii burkholzii at this point because she was still asymptomatic and negative, because if this hadn't been positive, we probably would have stopped here. But between day 33 and day 81, she developed abnormal sensations in her fingers, tingling, and very severe high headaches and migraines to the point that, you know, she was having a hard time functioning. At this point, you can see just what we've seen in some of the previous, and this is the reason we will always do PCR from blood and always do PCR from enrichment culture, is that she's now positive in enrichment culture, but negative from blood, and with exactly the same genotype that we got out of her earlier. What is striking, and we talk about seroconversion with Rocky Mountain spotted fever because it's such a pathogenic organism in a matter of 7 to 10 days. It was 128 days before we got a sample that we had an antibody titer above 1 to 64. At this point, once she was blood culture positive and seroreactive, she was treated by Chris Woods at Duke University Medical Center and had resolution of all her symptoms, seroreversion, which has been our experience. If you get rid of this organism, the antibodies go away and you're not able to detect it in the blood. This last example is an 18-year-old woman with peripheral visual loss, sensory neuropathy, and hallucinations of 4 years. So rather than the acute patient in the previous slide, this is a patient that presumably has been infected for a long period of time. Her progression was headaches, joint pain, back pain, mood swings, and dizziness, and obviously had been seen by numerous physicians, including a neurologist, psychiatrist, and neuro-ophthalmologist. And her results are on the next slide. So the story here is that we were not testing for Bartonella cholerae because we didn't know that humans were infected with Bartonella cholerae when she was entered into our IRB study. And we, as in Dr. Ricardo Maggi, who has done all the human testing in the BAPGM platform, was able to get a positive PCR result with both conventional PCR and quantitative PCR, but we could never get a sequence. Essentially, you've got to have enough template DNA to amplify up to a level that there's enough DNA that a sequencing laboratory can give you a sequence and say what the organism is. So we were really highly suspicious that this young lady was infected, but due to the fact that she was negative to Henselae, Winstonii burkholzii, and Quintana, which was the other organism that's not on this slide, we reported her out as negative because my IRB specified that unless we could put a DNA sequence on a result, we were not allowed to report these out as positive. So 294 days later, her mother contacted me again, almost a year out, because she was getting worse and her visual acuity had come down to the point that she could see straight ahead, but she couldn't see out to the side of her face on either side. And being a teenager, she had been told not to drive a car, but being a teenager, she continued to drive a car. So her parents were obviously extremely concerned with what was going on with their daughter. Here, we now went back and pulled her sample from day 0, and she did have an antibody titer to Bartonella Quintana. She had a higher antibody to Bartonella Quintana at this point in time. It was repeatable at day 507. She too was treated by Chris Woods at Duke University between day 294 and 507 with a couple courses of antibiotics to which she was not compliant. She would take them, not take them. And when she was proven to be reinfected or still infected on day 507 by enrichment culture, still had the same antibody titer her parents convinced her that she needed to be compliant, take her antibiotics, and ultimately she had, again, complete resolution of her visual problems, her headaches, her migraines, and her other mood swings, and total seroreversion. So just going from a couple individual patients to a couple larger studies, I just want to emphasize too, we started out with Bartonella because we felt veterinarians were at risk due to their exposure to vectors and arthropods. So you can look at this up here, but 198 individuals, 296 individuals. This was a single rheumatologist, Dr. Robert Mosiani in Rockville, Maryland. In regard to the male and the age and the female and the age, you'll see that both in Bob's cases and in the veterinary group, females are far overrepresented. What this means is anyone's guess, but certainly we know that in regard to some idiopathic autoimmune disease and rheumatologic disease, females tend to be overrepresented. And what you can see here that I wanted to emphasize on this slide is that in regard to seroactivity, realizing that a negative antibody titer might be more the norm than not, we ended up with 49% seroreactivity. Bob ended up with 63% seroreactivity using exactly the same antigen slides, and again, greater than 1 to 64 to any of the 5 organisms that we titer against. And if we look down here at the PCR results using the BAPGM enrichment platform done by the same investigator, Dr. Ricardo Maggi, in the same laboratory, which happens to be a biosafety level 3 laboratory, Ricardo actually got a higher prevalence of DNA in patients with rheumatologic disease. So again, emphasis on the fact that Bartonella causes chronic infections, emphasis on the fact that the Israeli physicians have documented a progression in a subset of cat scratch disease patients to rheumatologic manifestations, and clearly an area that deserves far more attention than it's received to date. So the only thing I want to emphasize on this particular slide is because of the relapsing bacteremia that we had documented in cats some 15 years earlier doing experimental infection studies, we started to suggest that people who were chronically ill and had a suspicion based on arthropod exposure or animal exposure submit 3 samples to us, generally obtained on alternate days. So we refer to that as a triple draw. And this particular manuscript by Beth Portulik in our laboratory as a PhD student statistically demonstrated that if we got 3 samples and went through the BAPGM platform, we were more likely to be able to confirm infection with Bartonella henselae and cholerae than if we had only previously gotten 1 sample or only got 1 sample during the time of the study. So just a little bit to finish up on DNA, since PCR is an absolutely amazing, revolutionary diagnostic modality. And when it's positive and it's done by a good laboratory, you can take it to the bank. They should have amplified and documented the organism they say they're detecting in your patient. But we've also have to emphasize that, like every diagnostic test, there's the good, the bad, and the ugly. The good is it's able to take and start with 2 copies of DNA and amplify that up to 3.5, whatever 10 to the 13th is, copies of DNA. It works for most type of samples. So I can PCR from a dog, a cat, a cow, a horse, or a human, and I can PCR from a dog's liver, spleen, blood, CSF. So it works for different samples. It should be highly specific and relatively highly sensitive under ideal conditions, and it's easily adapted and customized. Essentially, you could buy a thermocycler and do this in your garage. But the bad is it cannot detect which is not there. And we've emphasized this previously, this very low concentration targets. Essentially, the way to think of this is PCR is the best test we have to detect a needle in a haystack. And if you've got a bunch of needles in a small haystack, it'll work every time. If you've only got 2 needles and a haystack as big as a barn, there's no way PCR is going to detect those needles for you. It's prone to be variable depending upon the reagent, the design of the primers, the gene target, and the thermocycler that you're using. So there's a lot of variables that are incorporated into this that the laboratory has to be aware of and has to make sure there's consistency in what they're doing once they've validated their test. And it can be inhibited by a large number of substances, which I've just listed over here, those inhibitors and where their sources are. And so the ugly is, in my opinion, a complete lack of standardization across laboratories. So let's start with conventional PCR and qPCR first. And we're going to always start with the same amount because this is the maximal amount that we can extract DNA from in our DNA extractor. And we're always going to have, which you can see in red, the same 4 targets. So this could be Bartonella, this could be Borrelia, this could be Babesia. This is our pathogen target. And all this blue is host DNA. So obviously we're not just testing the organism, we're testing the organism because we're concerned about it being in our patient. And then once we go through our preparation step here, we're going to do PCR, which is qPCR conventional. So what happens if we have these inhibitors that we've talked about, or what happens if we have non-specific amplification because of all the host DNA, or what happens in regard to what's called primer sequestration is we really don't end up with any amplification at all, okay? We end up going from 1 copy to 2 copies, which we cannot detect in conventional or qPCR, or we end up with the primers annealing and not amplifying. So essentially a negative PCR result is reported out by the laboratory. But if everything works right, and if there are no inhibitors, then we end up with this 10 to the 13th number of organisms, which is much more target DNA, essentially we'll say Bartonella DNA, than host DNA, which we can now see by conventional PCR by running this DNA out on a gel, cutting out the DNA from the gel, sending it to a sequencing laboratory and getting it sequenced, or by qPCR where we can actually visualize on a screen the DNA being amplified, our target DNA that is. So ddPCR has been around for a decade or more, and it's been used primarily in cancer identification of low target mutations in association with host cells. So it was designed to overcome the limitations of conventional qPCR for that testing purpose. And what we're going to do again is start out with our same amount of sample that we can extract. We're going to start out with our same 4 organisms. And what is going to be incorporated is this step here, where the equipment essentially emulsifies all of this DNA, places it in individual droplets, which would be analogous to a flow cytometer, is what we're going to see ultimately. And then we're going to do the same thing we've done with conventional and qPCR. We're going to do a PCR reaction. But the difference now is that when this goes through our flow cytometer, it's going to throw out all of these negatives, which have no DNA. It's going to separate the host DNA, and it's going to separate our target DNA, which I'll show you here on a subsequent slide. So the power of droplet digital PCR is taking these low number of organisms, creating an amplification step, which we've talked about, some way of enriching this, but then by partitioning, we separate out our target from all the rest of this host DNA, which for us diagnostically is junk DNA that just makes our lives more complicated, okay? And so last year in the Journal of Microbiological Methods, Dr. Maggi and Dr. Miller at Galaxy Diagnostics successfully demonstrated that we could amplify 16 different Bartonella species. Those were the ones we had access to. And we could create co-infections with these species, both in the laboratory as well as in clinical specimens, and that we could detect these with a very, very high degree of sensitivity, which I'll show you on this slide. So this is from table 5 in the methods, and these are clinical specimens, 112 from 38 individuals that were suspected to have and did have Bartonella infection. And what's important here is that this is what we have done for years up until we combined BAP-GM enrichment with Droplet digital PCR. So this would be our historical standard, if you will, gold standard assay to which we've compared Droplet digital PCR. And you can see the results are dramatic. 53 detected out of 112 samples versus 6 detected out of 112 samples. And you can also see that there is still utility in regard to enrichment culture. We go from 20 being positive, 12 being positive, 11 being positive at 14 days, and 10 being positive. Obviously, the longer they're in culture, the trend is we're detecting fewer. So what I'd now like to show you on the next slide is the utility of this approach using BAP-GM enrichment blood culture and ddPCR. And what you can see here is this is table 5 from the Journal of Microbiological Methods manuscript with the validation. And these were 38 individuals that we referred to as suspected Bartonella cases, where they provided triple draws. So we have 112 samples that were tested from these individuals, and all were proven to be infected with Bartonella. And what is very obvious is the difference between qPCR with 6 positives out of 112 versus ddPCR positives, where we have 53 out of 112. What is also obvious is that enrichment culture still helps us detect some individuals that are not detected even by ddPCR directly from blood. So you can see here that we have a decreasing number from 12 to 11 and 10. But if we look down here, this slide tells us when was that case first detected by qPCR, or when was that patient first detected as Bartonella positive by ddPCR. So 3 in blood with qPCR, 14 in blood by ddPCR, 1 in a 7-day culture for the first time, 4 in a 7-day culture. None were found in the 14-day culture, but ddPCR actually detected an additional 6 patients infected in the 21-day culture. So this is an Excel from our laboratory form, and it's obviously all de-identified. Ricardo works solely with numbers. He has no idea what's coming in or what he's testing at that point in time. And if you look at the qPCR columns, you'll see that it's predominantly negatives. Predominantly negatives. This is a lot of testing. This is a lot of work. Again, blood, 7-day, 14-day, and 21-day. But if you look at the ddPCR columns, what becomes obvious is we start seeing a lot more positives. And what becomes also obvious when we look at individual patients reflected by these big arrows in the ddPCR is positive here, and positive here. And similarly, negative over here on 3 samples from blood by qPCR and ddPCR, but positive, positive, and this is, again, a different blood sample than this one since we're doing triple draws here. So obviously this is good news and bad news. The good news is we have further improved our ability of being able to say to a veterinarian or a physician that a patient has Bartonella DNA in their specimen. The bad news is this is not a user-friendly approach and requires 3 blood samples to do what we're doing on a research basis and extended incubation times to be able to document the highest degree of sensitivity with this platform. And the way this is, Ricardo gets these readouts for Droplet digital PCR, and these are spike samples with Henselae, Cholerae, and Bartonella Quintana. So if we start over here, our Bartonella is going to appear in this quadrant. Our DNA application occurs here. Host DNA is amplified here, and droplets that end up having both host DNA and Bartonella DNA will end up in this quadrant. So you'll see with the water control, there's no host DNA, so all 3 of these quadrants should have no drops in them whatsoever. If we put naive blood in that's not infected, that blood should have host DNA in it, but no Bartonella DNA or no DNA containing both droplets. And if we go to the remaining, you'll see that every one of these is amplifying Bartonella DNA, and everyone has droplets that contain both Bartonella DNA and host DNA. What I'm really happy about at this point in time is the specificity of droplet ddPCR seems to be somewhere between 99 and 100%. That's important because we cannot sequence at this point, based on the way the equipment is designed, the amplicons that we're generating like we could with conventional PCR and qPCR. So if we didn't have an extremely high degree of specificity, this would not be a modality that we would be able to use diagnostically. So what I hope I've illustrated is that BACGM helped us get the organisms up to a point that we could amplify it with qPCR and conventional PCR, but we knew based on the triple draw we were missing some patients. When we add in ddPCR partitioning with droplet digital PCR, that took us one step further in being able to enhance our ability to say Bartonella DNA, yes or no. And what we're currently working on is protein capture that Galaxy has already done and validated as a CLIA test for detecting Borrelia burgdorferi peptides in association with series in the urine of patients for the diagnosis of Lyme disease. We are now going through the process of using protein capture for Bartonella and comparing protein capture with ddPCR results to see which of these will be the most sensitive modality. So to conclude, advances in direct detection and Bartonella diagnostics. As we've emphasized repeatedly throughout these modules, we've got a large and diverse pet and wildlife reservoir in nature that periodically these organisms make the jump out of a reservoir into one of our patients. There are numerous vector-competent arthropods and some that we suspect to be vector-competent that are involved in transmission. There are numerous reservoir-adapted Bartonella species. The number keeps changing. And these organisms are capable of inducing persistent intradermal, intravascular, and tissue infections. They're difficult to detect or isolate diagnostically, and what will not be considered by me in these modules is I do not have an MD behind my name, but in dogs and increasingly in human, we're finding that they're as challenging or more challenging to treat than brucella infections, which are phylogenetically closely related. And with that, I want to thank you for your attention. And again, I hope that I've helped clarify what diagnostics are available for a patient with suspected Bartonellosis, what their advantages, disadvantages, and limitations are, and how enrichment of the sample is absolutely critical for our direct detection of these organisms regardless of the laboratory that's doing the testing. With that, again, I want to thank everybody in the laboratory and all the PhD students that have contributed to work with Bartonella. These 3 are currently in the laboratory working on this genus. And with that, I'll leave you with the references, and thank you very much.